So now that we've talked about bacteria, we're going to talk about how we can work with bacteria in the microbiology lab. To work with microorganisms generally requires these basic steps: inoculation, incubation, isolation, inspection, and identification. Let's talk about what's involved in each of these. Inoculation is where we take the sample from, for example, in a clinical setting from a patient and put it on the appropriate growth medium, something that contains the right nutrients, something that's in the right condition to grow that microorganism would be the inoculation stage. Now there's some terminology that comes with talking about this inoculation stage. When we say something has been cultured, it has been grown, right? Culturing would be the verb of going through the process of growing those microorganisms. The resulting growth would be called a culture. So you might see that word in several different contexts. The sample itself that was introduced to the growth media would be the inoculum. The act of moving the inoculum onto the media would be inoculation, right? And keep in mind, intentionally inoculating the growth media would be inoculation. Unintentional or accidental contamination is where you have organisms that are growing that you didn't intend to grow there. So, of course, this is where aseptic techniques are really important. We are trying to inoculate. We are trying to avoid contamination. We want to avoid contaminating our cultures. We want to avoid contaminating ourselves and the environment that we're working in. Typically, microbiology media comes in one of three physical forms. So liquid media is typically referred to as a broth. They're usually in these glass test tubes. They can have different nutrients added, different dyes added, things like that. But when something grows in liquid broth media, we're looking for evidence of growth would be something like turbidity, right? If it's cloudy, that's going to be an indication that bacteria grew. Now, not all of them grow uniformly throughout the media, and so sometimes you'll have growth that either is sort of like floating flakes. You might have something that sediments down to the bottom. You might have something that does what's called a pedicle, which is sort of like a waxy sort of scum that forms at the top of that pool of liquid. Some forms of media are considered semi-solid, which they're going to have a little bit of agar added to them, so they are not quite liquid, not quite solid. These are going to be important for testing things like motility. Do they have flagella? Can they move through the media? And you can see if they can move freely, you're going to see what looks kind of like a cloudy formation. If you look at the second tube here, that would be one where they inoculated the bacteria right down the center of that tube, but the bacteria did not have flagella, so they grew right along the inoculation path. They didn't move through the media. We also have very oftentimes agar that's added in a much higher concentration so that it's solid. Now, it's reversible depending on temperature. So agar is typically the special type of carbohydrate that we extract from a certain species of algae that's not digestible. So if it's got agar in it, it could be solidified at about 42 degrees Celsius, but we can liquefy it if we warm it up to 100 degrees. So this is a good way to inoculate for various purposes. We'll talk about the different types of media tubes that use a solid media. Types of culture media include what chemical composition they have. So if it's defined, it means it has precisely measured ingredients. The composition of them is absolutely known. We know exactly how many carbohydrates, which type of proteins, those types of things. Complex media is much more common in the microbiology lab because it has a sort of a random mix of, think of it sort of as general purpose media. It can pretty much support the growth of any microorganism. In the clinical setting, we usually have to use a type of complex media that's considered enriched, meaning it's going to have a mixed composition of nutrients, but then it's going to have some special growth factor that's going to help us select for certain pathogens. So when we say something is fastidious, it means it will only grow if that specific growth factor is present. So for example, blood might be added to a type of media if we're trying to look for hemolysis, right? Certain bacteria can rupture red blood cells when they're in, and that's usually one of the virulence factors associated with those infections. So if we're trying to grow those pathogens in the lab, we need to feed them what they want, which would require that special growth factor. Clinical settings, of course, use transport media quite a bit. So you don't want the sample you've taken from a patient to immediately start growing once it's placed in the media. You want something that's going to keep them alive but not allow them to grow. And that means when it gets to the lab and they start running tests, they're not looking at something that's overgrown artificially. Because bacteria, we haven't talked about their growth rates yet, but E. coli, for example, has a regeneration time of 30 minutes. So by the time the lab gets the sample, if it wasn't inoculated on transport media, within 30 minutes, you're going to think that patient has twice the bacterial load than they actually do. So some examples of enriched media are usually very colorful as well, some of the dyes that they add, but blood agar gets its color really from the blood that's added to it. So here's an example of those, for example, Streptococcus. There are certain species that are capable of hemolysis, and so when they break down those blood cells, you can see there's sort of a little halo around where the culture has formed. Chocolate agar sounds yummy, but there's no cocoa involved here. This is actually cooked blood, which gives it that brown color. But this is an important type of media that they use to diagnose gonorrhea, for example. Neisseria species grow best on chocolate agar. And then there's a disease called Legionnaire's disease, which is a respiratory infection that is specialized on this charcoal yeast agar. And so these are very definitive diagnostic tests that use these very special kinds of media that allow only certain things to grow, or at least it allows us to have more specific diagnostics. Selective media is that just what it sounds like. It's going to select for or inhibit some species. So if you're looking at this image here, it's probably nutrient agar, which is a complex media. It's got nutrients that pretty much support the growth of anything that was inoculated on it. But if you add salt or some other component to that media, you might actually inhibit the growth of certain things, right? So Staphylococcus would grow great on a type of media that had more salt than not. So this is selective media is going to be used in certain circumstances. Differential media is important because it'll allow us to differentiate those organisms that are growing on it. So for example, what's different about it is after it's grown on this media, sometimes the bacteria growing on that media will take up the dye from the background media. And so that's the case probably on the far right on this plate that you're looking at. Those colonies are sort of a red colored colony. The one in the center, it's taken up some of the dye, and the one on the left, it's actually changed the media behind it. So what used to be red is now a yellow color, and very oftentimes those color changes are based on pH changes, so we'll talk about that as well. Sometimes the difference is at the end of the inoculation and incubation period, you're going to see gas produced. So that would be something that helps us differentiate between different kinds of samples. Now, some media can actually serve both purposes, both selective and differential. So MacConkey agar is one of those. So if you look at the far left here, that's a nutrient agar plate. We've got two samples on it, E. coli and Staph aureus. And that nutrient agar plate is really important because it tells us that the cultures we're using are actually viable. Because if we inoculate those same cultures on MacConkey agar, you'll see that E. coli is selected for. It's actually capable of growing on that media, but there's not enough salt in that MacConkey agar to grow Staphylococcus. So you can see here there's no growth, and that would be the selective feature of this media. What you also see is a differential factor, right? Because if you look at the color of the colonies of E. coli when they grow on nutrient agar, they're just sort of a tan or clear color almost. When they grow on MacConkey agar, they actually take up the dye from the media that they're growing on and they put it inside their culture, inside their cells. And so those colonies actually look like a different color. So this is what we mean by being both selective and differential. in the case of the Salmonella species, they didn't show us the control there. We don't know what it looks like on a nutrient agar plate, but it clearly grows differently than the E. coli. So that's one of the differential characteristics that you could use this type of media. In some cases, we want to know their oxygen requirements. And so when you remove oxygen from a substance, you have reduced it. And so when we call what we call reducing media, the most common one is what we call thioglycolate media. And this is a type of media that is made in such a way that it excludes oxygen at a very regular rate. So by the time you get to the bottom of the tube, there's no oxygen. So if you were to inoculate this tube, and the growth at the end of the inoculation period was all at the top, so that's what these little black dots are. Those are representing the colonies that have grown only on the top of this tube. We would call that an obligate aerobe, meaning they can only grow in the presence of oxygen. If the growth was all at the very bottom of that tube, these guys would be obligate anaerobe. And in this case, it's not just that they don't require oxygen. Many times, if you're an obligate anaerobe, it's not that you don't require oxygen, it's that oxygen is actually toxic to you. And it might sound strange for something as necessary as oxygen can be toxic, but recall that oxygen can be converted into what we call free radicals. So we're gonna see in later chapters there's actually 6 forms of toxic oxygen, and if you don't have the right enzymes, you can't neutralize those types of oxygen, and it actually keeps you from growing. So that's what's going on here. If the growth is heaviest at the top, but you can see some growth that goes all the way down to where there's absolutely no oxygen, these guys would be called facultative anaerobes. So I usually interpret the word facultative as sort of can make do. And so in this case, we're saying it can make do without oxygen. It's not its preference, but it can actually still grow. It's just not going to thrive as much as it would when it gets oxygen. If it grows evenly throughout the entire tube, clearly oxygen has no impact on this organism. And so it gets the really long, obnoxious name of aerotolerant anaerobe. But this would be how we would characterize those organisms based on their oxygen requirements. Fermentation. Recall that bacteria, when they're fed carbohydrates or sugars, they're going to ferment and produce acids. And so very oftentimes the results of a fermentation media, if you provide the organism with a specific carbohydrate, if they can ferment it, they will produce acid. The acid will change the color pigment. So in this case, the pigment that's usually used here, the color indicator, is a pH-sensitive dye called phenol red. So if it's capable of digesting it, then the acid will be produced, and the acid will denature those protein pigments and turn it into this yellow color. Now, in addition, sometimes they're going to use what's called a Durham tube. A Durham tube, it kind of looks like a little miniature test tube that's been inverted and placed in this liquid media. And so any bacteria that are growing in that tube, as they produce gas, most of it escapes off the top of the media, but some of it will get trapped in that little glass tube, and so that will indicate that that gas was produced. So the combination of the color change and the gas production is going to be very indicative, and it'll help us identify which bacteria are growing. Now, once we've inoculated, we want to make sure that growth media is placed in the right conditions. Not all organisms can grow under the same conditions, both oxygen-wise, temperature-wise, even pressure sometimes can have an impact on how they grow. And so temperature is usually one of the most important characteristics, right? And if an organism is placed in an environment that's too high, think about what would happen to its proteins, right? So if you're a living cell, proteins are used for things like enzymes. Well, if you overheat those proteins, they will denature and those enzymes will no longer function. That is going to have a huge impact on their metabolism and their growth rates. The other thing that's going to be very susceptible to too high of a temperature is the phospholipid membrane of the cell membrane. So if you overheat that membrane, it's going to become so fluid that it's no longer going to be functional. Same thing goes for if the temperature is too low, right? Particles have to be moving around and bumping into each other for metabolic reactions to occur. So if the temperature is too low, everything slows down. So the growth rate is going to slow down. Same thing goes for the membranes. If your temperatures are too low, they're going to get so fragile because they're when those membranes are not fluid, they're going to be fragile and going to be susceptible to breakage. So temperature is really important for a microorganism in culture. In fact, we can classify them based on that temperature requirement. So the archaea are going to be the two on the far ends here, right? The psychrophiles, probably going to be those things that can live maybe in the Arctic Circle. Hyperthermophiles are going to be those that live in the hot springs. The thermophiles are probably those kinds of organisms that live in a compost pile that's properly maintained, right? Why compost breaks down organic matter is because the bacteria in there have the optimal growing conditions. Where we're really going to be most concerned are going to be mesophiles and psychrotolerance, because mesophiles, if you look at where that optimum is, it's about 37 degrees, which is human body temperature. So mesophiles are going to include not only the good guys that live in and on our bodies, but pathogens. Most pathogens are going to be mesophiles. Why we worry about something like a psychrotolerant, right? It can tolerate the temperatures of the refrigerator, for example. So if we have foods that are stored in the refrigerator for too long, those bacteria might actually, if they've contaminated that food, can be growing in that food. And then when you eat it, if you don't heat it properly to kill those bacteria off, you might be exposed to that. That's the case for things like listeria. It's an organism that can cause meningitis. Another organism that we worry about a lot in blood products, right? So blood bank products are stored in refrigerators, but the organism that causes the plague, for example, can survive at those temperatures. So we worry about psychrotolerance, even though they're not direct pathogens, based on where what their preferred thermal regimes are. It's because once they get into the human body, then they'll actually thrive, but they can tolerate those colder temperatures. Now, once we have cultures grown, because we've chosen the right kind of growth media and the right conditions for incubation, isolation is important, because if you run tests on organisms from a mixed culture, you'll never know which organism was responsible for the test result. So we've got to isolate species before we can get good data. And so it's rare to get a pure sample from, for example, a sample you pull off of someone's body, because we have a lot of different microorganisms. But if you're working in a lab and you've isolated those species, then you can end up with pure cultures. If we're in research conditions, of course, we can start with pure cultures, and hopefully if they're not contaminated, they'll stay pure cultures. In some cases, we have mixed cultures, and in this case, you can see it's a known mixed culture because it's evenly distributed, two different kinds of colonies that you can see on this growth. And by the way, if it looks different, it is different, right? So not only pigment, but size, the sheen on the surface of it, right? Some colonies are going to be sort of a matte, dull color. Some are going to be real shiny. Some are going to be fuzzy. If it looks different, it is different. So a mixed culture is probably a known combination of bacteria, whereas something that's contaminated, you're probably going to have the majority are going to look one way, and then you're clearly going to have sort of the outlier. So this white mass here is probably the contaminated portion of this colony, of this culture. And so in order to isolate, we have to use special techniques. The most common one is called the streak plate. So what's going on here is they take the initial sample and they spread it across just a small portion of the plate. They re-sterilize their loop, and then they come back to that plate and they drag a little bit of it from that original sample or that originally inoculated area and spread it out over the next area. They re-sterilize their loop, and they keep doing that over and over until they've covered the whole plate. And so what you're aiming for is at the end of the day, after incubation, we're going to have what are called colony-forming units, which are going to be individual colonies that are isolated from each other in such a way that you could take that one colony and then move on to the next step or the next test, and you would get very clear answers because you have an isolated colony. So basically what you're doing in the streak plate is just diluting the sample over the surface of the plate. The pour plate is another technique. This actually requires a serial dilution. So you've got a liquid broth type situation here where you've got a sample and then you dilute it at a very standard rate. You can do some math and basically back calculate and get some population data off of this as well. But this is a little more time consuming because as you do these serial dilutions, then you inoculate the plates. And so at some point you get to a point where those colonies are finally isolated enough that you can work with them. And then finally, what's probably the most user-friendly time-wise is the spread plate. And this is where they would take just a sample, put it into the plate, and then use these sterile spreaders, which they sometimes refer to as hockey sticks because that's what they look like, and just spread that sample right across the surface. And so you can do, many of these plates very quickly as opposed to those other two techniques that we saw. So this is very common in a clinical lab where you're doing lots of samples potentially. Now once we've got isolated samples, we can then inspect them, right? So when we're looking at them on the culture plates that we've grown, we would just look macroscopically. So what we just were talking about, the color, the sheen, the edging. So if you look at colonies, some of them are going to have a very regular, smooth outer edge of the colony. Some are going to have irregular. There's lots of different terminology we use to characterize those colonies. But more importantly, we can also put them under the microscope and go further with that inspection. And so by no means do you need to memorize all these numbers, but realize that when we start looking at eukaryotes like protozoa, algae, fungi in the form of yeast, or multicellular fungi like mold, you're going to see a range of sizes from 3 to 4 millimeters in those in the protozoa and algae. In the yeast, it's going to be a little bit smaller. When you get down to bacteria, now you're talking about the nanometer range through about 750 micrometers. Viruses, of course, are going to require electron microscopy because they're 1,000 times smaller in some cases. So most viruses fall in this range, but there are actually some that are really huge. In fact, there's one group that they named the Mimi virus because they thought they mimicked bacteria. In fact, they originally thought they were bacteria because they were so large. But for the most part, viruses are very small and they're only going to be seen with those electron microscopes. So again, don't memorize these numbers necessarily, but use it as a reference, right? If you're in the lab working on the microscope and you're asked to estimate the size of the organism that you're looking at using a mathematical calculation that we're going to be able to do, because you can't lay a ruler next to that bacterial cell and measure it directly, but we do have some math that we can do to estimate the size of the organism that we're looking at under the microscope. So if you can go back to this sort of reference list here and make sure that you're at least in the realm of possibility when you're estimating those sizes, it can give you a little bit of a double check. Now keep in mind, if you're in science, especially in medicine, the metric system is really key, right? As Americans, we are really accustomed to the English standard. So for example, the unit that we would use for weight in the English standard is probably the pound, as opposed to the metric system, which is the gram. If you were measuring volumes in the English standard, you would use pints, quarts, gallons, something along those lines, as opposed to the liter in the metric system. Of course, in this class, what we're going to be worried about are measurements. So distance, length, right? And so if you were talking about the English standard, you would probably use inches, yards, miles, depending on what you were measuring. But in the metric system, of course, it's the meter. And so meters are going to be the most important characteristic here. So why we use the metric system in science and medicine is because it's based on this unit of 10. And it's a lot easier to move from very, very large to very, very small by just moving a decimal point. If you can imagine trying to do the math and estimating the size of a bacteria using inches, you would end up with some enormous, incredibly small fraction of one, which which is really sort of hard to work with. So that's why the metric system is so useful, because you basically just jump by a unit of, or a place of 10. Now in this class, we're really only concerned with this part of the metric system. You should really be familiar with the whole system, but obviously the base unit here is going to be the meter. If you move down one level, you're at the decimeter. That's sort of an unusual unit that's not used very often. If you move one more decimal point, now you're at the centimeter level, and that's that is 1/100th of a meter. You can see on the far left here this exponent that goes with each of these levels. Make sure that you know those. Those are going to be user friendly. But realize that exponent, we're really saying you assume 1 times 10 to the -2, right? When it's a -2, you're talking about moving that decimal point to the left so that you're looking at a smaller number. Where you're going to, most of the conversions that you're going to be doing are going to be between centimeters, millimeters, and micrometers. Now keep in mind, when you went from centimeter to millimeter, you moved one more spot. There's a tenfold difference there, but there's really no useful units between millimeters and micrometers. And so you actually move 3 spots when you go from milli to micro. So realize that there's 3 decimal points there. And just make sure you're using the right symbol, right? The micrometer symbol is this Greek letter mu, which is sort of looks like a cursive U. So be familiar with jumping back and forth between these 3 levels. We'll get some practice with that in the lab. When we're using the microscope, we have to consider the resolving power, or what they call the limits of resolution. In our class, we're going to be using compound light microscopes, and you can see here's the range that we're limited to. We're anywhere from 200 nanometers to about 10 millimeters that we're going to be able to see using the microscopes that we have access to. The pictures in your book are primarily electron microscopes, so you can see it can go much, much smaller. But now there's actually microscopes available that can go way down to, they can resolve or have such high resolution they can see half the diameter of hydrogen atom. And so we have some really, they call atomic force microscopes. They're really incredibly powerful microscopes. We're going to be working here in this 200 nanometers to about 10 millimeter range with the organisms that we're going to be able to see. So here's the instrument that we typically use in our microbiology class. We're going to have, it's called a compound light microscope because we have 2 sets of lenses. We have the ocular lens, which of course is where you look through, and then the objective lenses. And we have 4 different objective lenses that are all attached to this, what they call the revolving nose piece. As you place your specimen on the mechanical stage, you lock it into these stage clips, and then you can move it around, and then we will rotate that nose piece and put the right objective lens in place, and then we can have basically a double set of lenses, and that's what they mean by compound. So this is a version of what we call a bright field microscopy. So when we're using visible light, we have to consider refraction of that light. So when light is traveling through air, it's going to be traveling without much hindrance, but as soon as it hits something with a different density, like the glass of the lenses, it's going to refract or bend. And so we use this to our advantage. If we understand light refraction, we can place those lenses at the right distance and the right shape and force that image to where it can be viewed. And so the convex lenses in the human eye look just like the ones we use when we have these microscopes. And so by having a convex lens like this, a double convex lens, it converges that focal point. And so the human eye, we can change the shape of that lens and put that focal point right on the retina so we can focus in on things. But what you notice here is the image doesn't just get magnified or enlarged, it actually also gets inverted. So keep in mind the human brain can reverse that image for you. You don't realize that what hits the brain is actually an upside down image because your brain actually processes that information and reverses it for you. But the microscope doesn't do that. So keep that in mind as we talk about how you're going to manipulate where you are on the microscope at any point in time. What you're seeing is actually everything in reverse, okay? Now keep in mind, because it's a compound light microscope, we have to take both sets of lenses into consideration. So the image that you're actually visualizing is going to be under what we call total magnification, because you have to take the power of the objective lens that's in place at the moment and multiply it by the ocular lens, which is usually always 10x power. And so you get this system here. This is the series of lenses that we have available, usually, and it's usually going to be printed right there on the sleeve of the objective lens. You can double check that these numbers are what you're using on the machine that you're working with. But the scanning lens, just what it sounds like, it's the lowest magnification, and it allows you to scan the specimen slide that you're looking at, so you can focus in on what you want very quickly. So if you take that, if that scanning lens is in place, you take that and multiply it by the ocular, and you have a total magnification of 40x. Now don't forget that x, that's the unit. Remember, in science, a number without a unit is really fairly useless. Once we get it in focus under the scanning lens, then we can move it up, rotate the nose piece and put the 10x lens in place. This is what we call the low lens, and this would give us a total magnification of 100x. Same thing with the high dry, we'd get up to 400x. And then ultimately we would, hopefully in this class, we get to use the oil immersion lens, which can give us the maximum total magnification on these machines. It's going to be 1000x. And of course it does require a drop of oil in order to take that last little bit of light refraction out of the picture, because the more it refracts, the more you lose some of the imagery. So we'll talk about that a little bit more when we get our hands on the microscopes. So keep in mind the reason why we need to know which lens we're working with at each time as well is not just for the image that we're seeing, but for understanding what we call working distance. This is the distance between the lens and the specimen that you're looking at. So you can see here that the 4 objective lenses that we have available, the lenses are longer as you go up in magnification, which means that your lens is going to get closer and closer to the specimen that you're working with. So it's really important that when you're working with one of these machines, in order to not damage it, that you are very careful about using, paying attention to what knob you're moving at any point in time. So it's only when you have the the scanning power, the 4x lens in place, that you can use that coarse adjustment. When you move the coarse adjustment knob, the stage can visually move. It moves very far, very fast. Once you get out of the 4x lens, you want to make sure you only use the fine adjustment, because you can actually put that lens right through the specimen and break the slide, scratch the lens, and do some damage. So keep this in mind. And of course, anytime you get on a microscope, you want to start with the 4x lens in place, because it gives you working distance to put your hands in and manipulate that slide and get it into position. When you go to put the machine away, you also want to put it back in the scanning lens in place, so that you have lots of room to remove that slide, and so that the next person that comes to use that machine also has it in the proper position. Field of view is essentially the circle of light that you see when you look through the microscope. And so this field of view, depending on what magnification you're in, if you're at the lowest lens, of course your total magnification was 40x. If you were to place this image in there, it would look like this. Let's say we get it in focus, we move up to the next level. As you increase magnification, the circle that you see is going to look like the exact same size. Your field of view is going to look to your eye, is going to look like it didn't change. But what we actually did was zoom in on the center of that field of view. If we zoomed in again, you can see how we zoomed into the very center. So the take-home message here is make sure that what you're interested in zooming in on is in the center of that field of view. And this is where we go back to that idea that everything is inverted when you're looking at these images. Because if what you're trying to look at is way up here on the periphery of this field of view, when you zoom in, it's going to be outside of the field of view, and you're going to end up having to chase that specimen around. And typically when you start chasing it, you go the opposite direction that you should, and you're going to spend a lot of time just looking for your specimen, when if it was centered before you moved up to the next level, you wouldn't have to chase it. It would still be visible. So the specimens that we view, especially in microbiology, are going to be prepared in what we call a smear. So we're going to take those bacteria in culture, take a very small sample, put it in a very thin layer across the surface of that glass slide. We're going to let it air dry to make sure it doesn't distort. If you put heat or blow on it, you're going to make those cells distort, and you won't get the proper data from that microscope slide. Then we're going to have to fix it, because when we go to stain these slides, they're going to go through several series of rinses, and they would just wash right off. So typically we're going to use heat fixing. We're just going to run it right through a Bunsen burner to denature the proteins in the cell membranes, and that allows it to stick to the glass. But you can also do a chemical fixing, but that uses acids to denature those proteins, but that also produces a lot of hazardous waste. So we tend to do heat fixing. Then, of course, we need to add some contrast, because these the cell structures are literally invisible under the microscope. So most microbial stains are going to be either in the family of salts, and if you recall the definition of a salt, it's something that dissociates into a cation and an anion. Depending on the different dye that's being used, one of those two ions is going to be then tagged with what they call a chromophore, or the pigment. Acidic dyes are going to be like opposites attract. They're going to stain alkaline structures, and basic dyes are going to stain acidic structures. Now, most commonly, we're going to use basic dyes. So think about all the acidic structures on every living cell when you think about the cell membrane. You have the phospholipid membrane, the fatty acid tails are going to take up pretty much any basic dye that you put on them. So we're going to look at these three different groups or general types of stains. A simple stain, just what it sounds like. It's only going to have one type of dye used, but these are really sort of limited in use because all the cells present are going to take it up in their cell membranes. They're all going to look exactly the same color. It has limited use. It can tell us a few things. It can tell us presence, absence of the bacteria. It could tell us population size. It can tell us morphology and arrangement. But really, if we want it to be more user-friendly, we need to take it on to a different type of staining. So we've talked about the Gram stain in the last chapter, and so again, this is going to help us differentiate between the two types of Gram status cells. So if you recall, which ones are the purple are going to be your Gram-positive cells, and the pink or red are going to be the Gram-negative cells. So we went through the stages of that, of the different steps required for the Gram stain back in chapter 3. A special kind of stain that really differentiates Gram-positive cells that have that waxy component, you recall we talked about mycolic acid. These are going to be called what we call acid-fast cells. The Ziehl-Neelsen stain was the historic name for this, but acid-fast bacteria, if you get a culture back from a paint sample and they have acid-fast rods, that are those that have this mycolic acid, and it's just a special kind of fatty acid that makes these bacteria very resistant to certain chemicals and the dyes associated with these staining procedures. In fact, to get that dye past the mycolic acid layer, you have to have a steam and heat added to these, acids added to this staining procedure to get that dye past that layer. What it does for the organism physiologically is it keeps them from drying out. So this is one of the reasons why it's so difficult to deal with tuberculosis, because that bacteria is quite viable outside of the host cell because it has this mycolic acid in it. We also talked about endospores back in chapter 3. So recall, these are very hardy, very thick, difficult layers to get past. So this one also requires a high level of heat to get these stains past that spore coat. This one uses a green dye called malachite green, and then we also have to counter stain the cells with safranin. The same dye we use at the end of the Gram stain can be used to make sure that the sporangium part of the cell is also visible. The capsule stain is called a negative stain because actually it uses two kinds of dyes. It uses an acidic dye that actually is repelled by the cell, and it sticks to the glass. It sticks to the background of the slide. The basic dye is then added, and again, very oftentimes they'll use either a red dye called eosin, And then the acidic dye is something called nigrosin. That black dye that's on the background on the right side there is probably a nigrosin stain. But basically what they do here, it's got a lot of advantages over the other types of stains. Number one, it allows you to see the capsule. So recall that the thickness of that capsule is sometimes diagnostic. So one of the big advantages here is because this type of staining doesn't require any heat fixing, because you take the sample and you actually add the dyes to the liquid broth with the bacteria, and then you just take a sample of that that's been mixed and place it on the slide and allow it to air dry. So there's no distortion. So you can see the morphology and arrangement is really easy to see on these, but also the thickness of that capsule can be estimated. And that's sometimes the way we distinguish between different bacteria, is just how thick the capsule. If there's a capsule present, you may hear that it's referred to as capsular or encapsulated. All of those mean that a capsule was detected, basically. Flagellar stains. So again, if you want to see the flagella, this is actually a very incredibly small structure compared to the other parts of the cell. So to give you some reference, if you could pick one hair on a human head compared to the rest of the human body, that would be the relative size difference between a flagella and the bacteria that has that flagella. So it actually requires a series of steps where you put a stain and then a mordant and a stain and a mordant, and you basically layer and layer and layer and to thicken up that dye layer on the flagella to the point that it's visible. And each of those steps is very pH sensitive. So this is a really tricky procedure. The stain that they use is usually called carbofuscin or Leifson's dye. And so you can see here, and again, we have two types of flagellar arrangements obvious here. The one on the left, again, a single flagella coming off one end, we would call that monotrichous. And the one on the right, we would call peritrichous, many flagella coming off all around the periphery. Fluorescent stains use both a dye that is fluorescent in nature and that is tagged to an antibody. And so antibodies have a very high affinity for the antigens they are matched to. And so really when they use a fluorescent dye stain technique, you're really sort of almost just seeing like a halo around the organism, because the antibody just sort of takes the shape of defines the shape around the organism. This is actually one way that we diagnose both rabies and syphilis. Those are both diagnosed using fluorescent stains because we have antibodies that can tag those organisms. This picture on the left is actually just a eukaryotic cell where they've used very specific fluorescent stains and antibodies to tag some of the proteins that make up the cell membrane, the nuclear envelope, and the cytoskeleton. So you can see that high detail associated with fluorescent stains. But of course, the best detail you can get with electron microscopy. So this actually requires what I always kind of think of as a little spray tan. So they take this very fine, heavy metal. Usually it's a combination of gold and palladium and some other heavy metals, and they basically spray it across the surface of the specimen and then bounce electrons off of it. And so you can get really incredibly highly detailed specimens. So when you look at the pictures in your book, some of the micrographs that you see, if you read the caption and it says TEM, that's a version of electron microscopy called transmission electron microscopy. And this requires actually sectioning. So they would take the specimen and slice it up into really incredibly thin pieces, and then the electrons would actually be sent through the specimen. And then the differences in density would be detected by the computer. If it's ever a TEM image that is actually in color, that's artificial. They have to go in after the fact and artificially colorize those pictures. Here's, again, a scanning electron microscopy. You're just literally scanning the surface of the organism. Here's a diatom. We talked about how symmetrical those single-celled algae that have very silicate cell walls. Here's an aspergillus fungus. So you can see those spores and the hyphae that they come off of. Here's one of your ciliates. There's one of your paramecium. You can see all the individual cilia. And streptococcus. So again, very high, fine detail associated with those electron microscopy images. Then finally, once we've isolated it, we may have taken some visual data on the microscope. Now we're going to use a combination of all those pieces of information to identify the species that we're dealing with. So some of those tests that we talked about, or the types of media we talked about earlier, maybe fermentation media, might use sugars, or we could use something like citrate and just see if the organism has the right kind of enzymes because of its identity, and its identity is based on its DNA. Its DNA determines the proteins. Proteins are enzymes. So if you have the right enzyme, you can use the right substrate. That can be indicated as a biochemical test, and we can use that data as part of our identification process. Sometimes we're not looking at the actual fermentation process, but we're looking for other waste products that they produce. So again, special media is usually going to help us get these answers. Now in the clinical lab, they typically aren't going to do these tests one by one. They're going to use what they call multi-tests. There's one called the E-test. This Entero tube, it can do dozens of tests all at once, one incubation period, very quick results compared to doing some of these other tests one by one. But that's really important in a clinical setting where you're trying to diagnose an infection. When you look at the data that's presented to you, this was a battery of tests that were run in the lab. The lab does not know what series of questions you're going to be using the results for. So they give you the answer to every test they ran. Some of these tests you'll need, some of them you won't. So keep that in mind. Don't ever just follow the data that you're given. You follow the map. So for example, always start with the first question. What does our data tell us about the gram status? So in this case, we have a gram-negative cell. So what we're wanting to do is to follow and move on to question number 2. Question number 2, is it rod-shaped or non-rod-shaped? Well, this is where you have to come into a dichotomous key with a little bit of pre-existing knowledge. So what is the word we use for rod-shaped? We use the word bacillus. So in this case, a bacillus, we want to go to question number 3. Can it tolerate oxygen? Can it not? Well, in our data, we're told that it is an aerobic bacteria. So we want to go to question number 4. Lactose, can it ferment lactose or can it not? And in this case, look at the symbol that they put, citrate negative, positive. So in this case, lactose positive. That means it can ferment lactose. So we need to go on to question number 5. Can it use citric acid as a carbon source? So here's where, again, you have to sort of sometimes make some judgment calls or some educated guesses. Citric acid and citrate are going to be the same test. Okay, so the lab and the dichotomous key may not use the exact same terminology. So in this case, citrate negative, so we're going to have to go to question number 8. So now keep in mind, the map gives you other questions. We do have data for the hydrogen sulfide test, but we don't need it. We have data for the acetylene test, but we don't need it. The test, or our answer, told us to go to question number 8. The gas from glucose, so we're going to see glucose gas on our data list. It says positive. So our answer is Escherichia. We've just keyed out this unknown organism. This is basically what we did. We followed this little path. So sometimes you're actually going to see dichotomous keys laid out like this as a flow chart as opposed to a series of questions. It's really the same information. It's the same way that we got to the right answer, right? So in this case, again, we have mapped this out or keyed this out to Escherichia. Now, very oftentimes you're also going to be asked for the definitive test. The definitive test is always the very last one used because it's the thing that distinguishes that organism from its closest relative. Right? In this case, the closest relative is Shigella. So the definitive test would be the glucose gas test. Right? And what do we mean by distinguishes it from its closest relative? Well, if you back up through the map, basically if the answer were Shigella, every answer along the way would be exactly the same as Escherichia until you got to the gas glucose. Or glucose gas question, right? That makes it definitive. Now, every test we used along the way was definitive, but when they ask for the definitive test, they mean the very last one. But that also means that anything we didn't use is considered a non-definitive test. So this is where when you're using a dichotomous key, it's very helpful to not only mark on the key which choice you made to move on to the next question, but also go to your data list and your lab results and check things off as you use them. Because if you've done that along the way, if you're asked for the non-definitive test, in this case, the two non-definitive tests were hydrogen sulfide and acetylene. The glucose gas was the definitive test. So keep that in mind. That's how you're going to use these dichotomous keys. This is really the process that you're working up to for the lab practical. And so your unknown ID lab exercises, you're basically going to be collecting data all semester long so that you can do exactly this on those unknown specimens that you're working with this semester. But keep in mind, you're going to be practicing this along the way. Every test, you'll have unknown specimens to key out on the test. Now, I said this was a very simplified version of things. Typically, when you get lab results, you're going to get dozens of lab results, not just the 6 or 7 that we have used here for an example. When you're using a dichotomous key, there's usually, 20 pages worth of a dichotomous key. We're going to use at least, I think it's like a 4 or 5 page key that we're going to use when we do the lab practical. On the test, you'll use a 1-page one, but it's still going to have 15 to 20 questions on it. So keep in mind, this is a very simplified version of what you're going to see. But keep that in mind. This is a process. This is a skill that you need to work on to make sure that you understand there's a practice set of organisms that we'll work with as well, so that you can get a little practice before the test. All right, that's it.